Hi, Assalamualaikum. So today we are presenting to you about gravimetric analysis and its application. Our group consists of 4 members, which are MAIZATUL, AKALIL, ZAINUDDIN, and FIRDAUS. Introduction. A quantitative technique used to precisely ascertain the amount of a certain chemical compound in an aqueous solution is known as gravimetric analysis. The appropriate component is selectively precipitated in order to perform a gravimetric analysis. Gravimetric analysis is used in elemental analysis of inorganic compounds, elemental analysis of ores and other industrial materials, and equipment calibration. It is also used in many industries to examine finished products and raw materials and ascertain the chemical makeup of alloys, rocks, and minerals.
There are 4 types of gravimetric analysis. First is precipitation method. The analyte transforms into sparingly soluble precipitate, which is then filtered and purified to provide a product with a known composition that is stable through appropriate heat treatment. After that, the product is weighed. Second is volatilization method. At appropriate temperature, the analyte or decomposition product volatilized. Next, the volatilized product is gathered and weighed, or else the product weight is inferred indirectly from the sample's weight loss. The third is electrogravimetry and thermogravimetry.
Basic steps in gravimetric analysis. First, solution preparation. Obtain a representative sample and, if necessary, prepare it for analysis. This may involve grinding, homogenization, or other sample preparation techniques. We also need to eliminate interfering materials in this process. Second, precipitation. A solution containing the analyte, which the substance to be determined, is treated with a reagent that forms a solid, insoluble compound with the analyte. This solid is called the precipitate. Third, digestion. Keeping the precipitate to remain in the mother liquor or solution it precipitated from for a certain amount of time in order to promote nuclear densification. During digestion process, small particles dissolve and larger ones grow. Larger crystals that are easier to filter out of solution are produced as a result of this technique. Fourth, filtration and washing. The precipitate is separated from the remaining liquid by filtration. This involves using a filter paper or other porous materials to capture the solid while allowing the liquid filtrate to pass through. The collected precipitate is washed to remove any impurities or excess reagents that may be adsorbed on its surface. Fifth, drying. The washed precipitate is dried to remove any remaining water or moisture. This is done by heating the sample to a constant mass. Sixth, weighing. The mass of the dried precipitate is determined using a sensitive analytical balance. Chemical factors and gravimetric factors are the methods used to calculate the amount of analyte in the weighing form. And last but not least, calculations. The mass of the precipitate is used to calculate the amount of the analyte in the original sample based on stoichiometry. Here you can see the formula for chemical factor and gravimetric factor.
Next, we talk about advantages and disadvantages of gravimetric analysis. So first, for advantages, no calibration needed. Second, result is calculated directly from the experimental data obtained. And third, high precision and accuracy. This is because it requires only an accurate analytical balance. For the advantage, first, time consuming, which is long experimental hour. Second, minor misstep will not produce a pure sample. Any impurities introduced during sample preparation, precipitation, or filtration can lead to inaccurate result. Third, need higher concentration sample. Higher concentration of the analyte can lead to more significant mass of precipitate that result in more precise measurement. Application of gravimetric analysis in pharmaceutical industry. Gravimetric analysis find various applications in the pharmaceutical industry, primarily in the determination of drug purity, the content of active pharmaceutical ingredient APIs, and the identification of impurities. The following are some specific applications. First, drug purity testing. Gravimetric analysis can be employed to determine the purity of pharmaceutical compound. By isolating and weighing specific component, analysts can assess the percentage of the active ingredient in drug. Second, quality control. Pharmaceutical companies use gravimetric analysis as a quality control measure to ensure that the formulation meets specified standard. This can involve determining the amount of a particular substance in a sample. Third, residue analysis. Gravimetric method can be applied to analyze residue or contaminant in pharmaceutical product, ensuring that the final drug is free from impurities that could impact the efficacy or safety. Fourth, formulation development. During the development of pharmaceutical formulation, gravimetric analysis can assist in optimizing the composition to achieve the desired characteristic and concentration of active ingredient. And lastly, dosage from testing. Gravimetric technique may be used to verify the consistency and accuracy of dosage from such as tablet or capsules, ensuring that each unit contains the intended amount of active ingredient.
Now we go to the methodology. First is selection of precipitating reagent. Choose a reagent that reacts selectively with the drug of interest to form a stable and easily filterable precipitate. The reaction should be stoichiometric to ensure accurate determination. Next is sample preparation. Extract or isolate the drug from the pharmaceutical formulation. This may involve dissolution extraction or other sample preparation techniques. After that is precipitation. Add the selected precipitating reagent to the drug solution. After that, digestion. Keeping the precipitate to remain in the mother liquor or solution, it precipitated for a certain amount of time in order to promote nuclear disinfection. Next is filtration and washing. A precipitate forms, which is then separated from the solution by filtration. Wash the precipitate to remove any impurities or excess reagent. After that is drying. Dry the washed precipitate in a drying oven to a constant mass, ensuring the removal of any remaining water or volatile substances. After that is weighing. Weigh the dried precipitate using the analytical balance. And lastly is calculations. Calculate the concentration of the drug in the pharmaceutical formulation based on the mass or precipitate of the stoichiometry of the reaction.
Now let's take a look at example of result. The technique is consider the determination of a specific drug X in a tablet formulation using gravimetric analysis. Suppose drug X react with a precipitating reagent such as silver nitrate to form a stable precipitate such as silver drug complex. Below are the example of the reaction. For the first one is precipitation. Add silver nitrate to the drug solution and a precipitate of a silver drug complex form. Next, filtration and washing. Filter to separate the solid from the liquid. Wash the precipitate to remove any impurities or excess silver nitrate. Next is drying. Dry the washed precipitate to a constant mass. Then weighing. Weigh the dried precipitate using an analytical balance. The last one is calculation. Calculate the concentration of drug X in the tablet formulation based on the mass of the precipitate and the non-stoichiometry of the reaction. In conclusion, gravimetric analysis is an effective and precise quantitative approach that is essential to the pharmaceutical industry. It ensures the quality, safety, and efficiency of pharmaceutical goods by determining drug concentration, detecting contaminant, and guaranteeing content consistency within dosage form. The approach entails precipitating the targeted component selectively, separating it from the formulation, and weighing the precipitate to calculate the concentration based on reaction stoichiometry. The precision and sensitivity of gravimetric procedures are critical, contributing to regulatory compliance and patient safety. This adaptable method not only examines dosage form accuracy, but also identifies impurities and validates the uniform distribution of active ingredients. Because of its broad impact on drug research, production and quality control, gravimetric analysis has become a vital tool for pharmaceutical quality assurance.
And these are the references that we use for this assignment.